Protocells are thought to be composed of genetic polymers, different reaction networks encapsulated in compartments. Uh, the presence or the ubiquitous presence of membrane-bound compartments in contemporary biology, the presence of different amphiphils on prebiotic Earth, and their ability to assemble large cell-like structures highlights their role in the emergence and evolution of protocellular life form on early Earth. Protocell membrane have been considered to be simple and composed of single chain amphiphils like fatty acids, fatty alcohol, etc., whose synthesis have been shown to be possible under pertinent prebiotic conditions. And towards this, fatty acid have been studied extensively because of its ability to form bilayer and large vesicular structures when the pH of the surrounding is near to the pKa of the head group of the fatty acid. However, if the surrounding pH is high or low, then the pKa of the head group it assemble into micelle and droplet-like structures respectively, both of which fails to encapsulate aqueous volume in them. What I was interested to look at is that what model protocellular membrane are composed of in terms of its composition and how the composition manifest or influence the physicochemical properties of the membrane. And where did the first protocellular life form emerge? As the composition and the physicochemical properties of the protocellular membrane would be largely governed by means of complex interactions with its physicochemical environment. Towards this, terrestrial geothermal pool has been considered one of the potential site for the emergence of protocellular life form, as different conditions such as day-night cycles or geyser activities, thermal evaporation, and seasonal fluctuations can lead to a geological phenomenon or geological setting known as wet-dry cycle in this condition. So wet-dry cycles have been shown to be facilitate the formation of biopolymers on prebiotic Earth, and this phenomenon, this wet-dry cycling, this setting has been studied extensively in literature, has been shown to facilitate the formation of RNA esters and peptides. And given the importance of wet-dry cycles on prebiotic Earth and the formation of protocellular life form, we wanted to study whether model protocellular membrane of different composition were able to sustain or survive under multiple wet-dry cycle, and how multiple wet-dry cycle can influence or impinge on the self-assembly process and the physicochemical properties of the model protocellular membrane systems. Towards this, we used a C18 based membrane system where we used 4 different compositionally different membrane system. One containing only oleic acid, 2 binary system containing oleic acid either with oleyl alcohol or with the glycerol monoester of oleic acid, and the tertiary system containing all 3 in 4:1:1 ratio. All those 4 systems have been previously studied, and the physicochemical properties have been reported in the previous studies came out last year from our lab. So when we subjected this 4 different membrane system to wet-dry cycling conditions and then took out different samples and analyze them. So when present in aqueous solution suspension, the amphiphils form this large vesicular structures, and when dehydrated in the dry phase, they first go to the gel phase and then form this multilamellar structures, and upon rehydration they again reassemble into vesicle-like structure. So once after different wet-dry cycle, we take out samples, observe them under microscope. We observe that all 4 different membrane system of different composition were able to form reassemble into vesicles even after 12 wet-dry cycles. The scale bar here is at 10 micron. So this large vesicular structures. However, this confirms that the systems were able to sustain wet-dry cycles, but it miss out the information about what happens on the on the physicochemical properties of the membrane under multiple wet-dry cycles. To look at that, we used a different solvatochromic probe. We first used Nile red, which is a hydrophobic solvatochromic probe. So upon excitation, when Nile red is present in water, its emission maximum was found to be at 660 nanometer. When Nile red get encapsulated in hydrophobic environment, for example in membrane, its emission blue shifts. And for example, changing in the hydrophobic environment, for example, membrane to droplet, it emission further blue shifts. So we use this ratio, intensity ratio of 16 by 660, and plotted that with a for all membrane system with different wet dry cycle. So this ratio 16 by 660 intensity ratio was found to be highest and lowest in presence of oleic acid droplet and micelle respectively, and was found to be intermediate in presence of 4 different membrane systems. So when we plotted the 610 by 660 ratio for all 4 different systems across different wet dry cycle, we observed that for uh, only in case of the binary system containing oleic acid and oleal alcohol, the ratio changes after the second wet dry cycle. The ratio is found to be increased, which indicates the decrease in the micropolarity or the water accessibility on the membrane. However, for all 3 other membrane systems, we didn't observe any changes in the micropolarity of the membrane. Next, to understand this further, we used another solvatochromic probe known as Pyrene. So when excited, Pyrene provides this 5 characteristic peak between 370 to 390, and the ratio, intensity ratio of peak 1 to peak 3 provides the information about the micropolarity of Pyrene. So this ratio is found to be highest when pyrene is present in water, and upon increasing the hydrophobicity of the microenvironment, the ratio decreases. So when we plotted the ratio for all 4 different systems across different wet dry cycle, we observed this interesting phenomena where we found that all 4 different membrane systems segregates into 2 categories, in which there's oleic acid. In one, there's with oleic acid and the binary system containing oleic acid and oleal alcohol, which has similar micropolarity, which is polarity, and the other containing oleic acid with GMO and the tertiary system. So what we observe is that whenever this GMO is present in the membrane, we observe the increased water accessibility in the membrane. So this could be boiled down to the higher or bulkier head group of GMO when present in the membrane. The membrane experience more water on it. So when comparing the peak 1 by peak 3 ratio, we observed only in case of the binary system containing oleic acid and alcohol, we see a decrease in this ratio, which indicates a significant increase in the membrane micropolarity. So this ratio polarity. So this indicates that there's a change that is happening when different membrane systems are subjected to wet dry cycles, and thus indicating that all the different components are not stable under wet dry cycle, and thereby reflecting on the manifesting on the physical changes on the physicochemical properties of the membrane under wet dry cycles. To look at the chemical stability of the amphiphiles, we first look at the net bilayer content in the solution after different wet dry cycle, and we observe that in case of oleic acid, there's a significant decrease in the bilayer content after 8th wet dry cycle. However, in case of oleic acid GMO system, panel C, there's no changes in the net bilayer content. However, interestingly, in case of panel B, which is oleic acid and oleal alcohol mixed system, we see the bilayer content started decreasing just after second cycle of wet dry cycles. So when we looked at the stability of the molecule, when we done the suspension on a TLC plate, we observed that GMO and oleic or GMO and oleic acid were stable even after 12 wet dry cycles. However, the band corresponding to oleal alcohol was absent after four wet dry cycles, so which indicates alcohol was not present in the system. So absence of further degradation band on the TLC plate confirmed that there's no chemical degradation of oleyl alcohol. However, the oleyl alcohol got evaporated out of the system under wet dry cycling condition. So the binary system containing oleic acid and alcohol got converted into the homogeneous system of containing only oleic acid, and the tertiary system eventually got converted into binary system containing oleic acid and GMO after multiple wet dry cycle, which was reflected on the changes on the micropolarity of the membrane upon subjected them to different wet dry cycles. We next looked at the changes in the vesicle size under multiple wet dry cycle and observed that in case of three system, or where it is oleic acid, the binary system containing oleic acid GMO, and the tertiary system, there's a decrease in the vesicle size upon subjected them to wet dry cycle, and which was observed just after the first cycle. However, this phenomena was found to be absent in the oleic acid and alcohol mix system. And when we looked at the encapsulation, how encapsulation efficiency changes upon multiple wet dry cycle, we observed that in case of all four systems, the encapsule efficiency increases with increasing wet dry cycles, but the pattern was different. Whenever there's GMO, or the glycerol monoester, present in the system, we observed that there's a sudden increase in the encapsulation efficiency. However, in case of oleic acid and oleic acid oleic alcohol mix system, the increase in the encapsulation efficiency is gradually. We used calcine in these studies to look at its encapsulation efficiency, which is a polar fluorophore. And to summarize all this study, we looked at the physicochemical properties and how it changes of this membrane and how it changes. So wet dry cycle was found to be impinge on the membrane composition and thereby changing the membrane physicochemical properties. So this work have been published recently in Chem System Chem. So I encourage whoever is interested to look at this paper to find it out and have a look at it. And with this, I would like to thank the people who are associated with this work and the funding agencies for their support. Thank you.